Good evening, ma'am.
Hello and welcome to today's immunology live session. I'm just waiting for others to join. So this week one of this course has been live, and I hope you have been going through the lectures. So in these live sessions, what we will be discussing uh, is that some basic concepts that will be discussed in the particular weeks. Like for example, today we will be discussing the concepts and some questions also of week one. And so this will be an interactive session, so you all will be able to ask me questions and I can answer. And the thing is, we will discuss some of the basic concepts which will be discussed in this particular course. I hope my screen and audio is clear to everyone. Okay, so I think I can begin. So, to introduce me, I am Surasree Pal, and I am a Prime Minister's Research Fellow working in the Department of Biotechnology in IIT Kharagpur. And in this course, I am this teaching assistant in this immunology course, which is taken by Professor Shudeep K. Ghosh and Professor Agnew Ganguly of IIT Kharagpur. And just to introduce you to this live sessions, I know there is another one of my colleague who is taking another live session in this course itself in the on the Fridays, right? And I'll just like to ask if any of you are or have also attended that live session.
Yes.
Okay, all right. So we will try to both of us, we will try to answer some of the questions that you ask in the blog blog of this course which is going. So we check the blog, and if you have any questions, right, so if you have any questions, you can post there, and those questions we can discuss in the live session, right? And also I'll be discussing some of the some questions which are relevant to this week's topic. So I think we can begin, right? All right. So I know that in another live session, and also in the this week's content which has gone live, you have gone through the basics of immunology. Now, I would assume that you all are naive to immunology. That is, like you haven't had any exposure of immunology previously, and in that case, um, like you must now after going through this course, like week one's content, you may understand that immunology is talking about this system of immunity that goes in our body to protect us from several pathogens, right? Right. And the thing is, what are our weapons in this immune system? So all right. So I'll just be introducing you to these immune cells, the cells which are responsible for providing immunity in our body. And the cells, the mother of all these cells is the pluripotent hematopoietic stem cell, right? So this pluripotent hematopoietic stem cell, there are two terms here. One is hematopoietic stem cell, which is HSC, abbreviated as HSC, and another is pluripotent. So anyone has any concept of what is a pluripotent cell? Can anyone share?
Pluripotent is basically a multiple cell kind of generation capacity. Like from that mother cell, it can be generated like many different kinds of cells.
Actually, right. You are quite right in this case. That is, pluripotency actually refers to this fact that one particular type of cell, yes, able to differentiate into different kinds of cells. Right. Yes. Shayan also, you're right. Shivam also, you're right. Right. So pluripotency basically means that this particular type of cell can differentiate and form different types of cells from one particular cell type. And this hematopoietic stem cell is the mother cells of all immune cells. So this cell usually resides in the bone marrow, where it first undergoes differentiation into two particular lineages of immune cells. So one lineage is, here you can see, the lymphoid lineage. So this HSC undergoes division and differentiation to form two lineages of cells, the lymphoid lineage and the myeloid lineage. Right. And this thing, remember, this is occurring inside the bone marrow. And from the common lymphoid progenitor, we will next see like what kind of cells can originate from common lymphoid progenitor. And the other type is the common myeloid progenitor, from which again differentiation arises different types of some progenitor cells. So first of all, why are they called common? Because they are the progenitor of multiple types of lymphoid cells or myeloid cells. Now from the common myeloid progenitor, as you can see here, the granulocyte macrophage progenitor and the megakaryocyte erythrocyte progenitor. These two types of cells originate and differentiate from the common myeloid progenitor. And from this granulocyte macrophage progenitor, as you can understand from the name of the cell, that the granulocytes and macrophages will be differentiated or will be produced or will arise from this granulocyte macrophage progenitor. And from the megakaryocyte erythrocyte progenitor, you can understand that megakaryocytes and erythrocytes or erythroblasts will be produced. So megakaryocytes are the cells which will produce platelets and erythrocytes are the RBCs, right? So from this common lymphoid progenitor and common myeloid progenitor, next we will see what types of cells again arise. So from the common lymphoid progenitor, you can see here the lymphocytes arise. The lymphocytes. Right. So you know what are the different types of lymphocytes, right? The B cell, T cells, and there are two other types of cells which are the NK cell and ILCs. ILCs are innate lymphoid cells. So what is innate, what is adaptive, we will learn later on. All right. So so from the common lymphoid progenitor, these types of B cell, T cell, NK cell, they all originate. And here you can see one thing that this is still taking place in the blood, right? The common lymphoid progenitor is originating different types of cells. From the myeloid lineage now, the immature dendritic cell from the common myeloid progenitor, one immature dendritic cell is originated. Also from the granulocyte macrophage progenitor, as I was telling you that this granulocytes, all types of granulocytes, for example neutrophil, eosinophil, basophil, they are produced. And also monocytes, right? Now and from the megakaryocyte, these platelets are produced which are important for blood clotting, and from the erythroblast, the erythrocytes or the RBCs are produced, right? And one interesting thing that you can observe here is that from the bone marrow, you can see when the cells are going towards the lymph node, they are being differentiated again to form some activated cells, right? So they are undergoing differentiation in the lymph nodes. And this, suppose in here you can see these B cells, T cells, NK cells, they will attain more and more differentiation and more maturity in these lymph nodes. Whereas in the tissues, the macrophage, the most important resident immune cell of the tissues is macrophage, which will be differentiated in the tissues, whose differentiation is taking place in the tissues. And this is the last and the final part of this immune cells, maturation of immune cells, that is from the lymphocytes, from the B cell, T cell, NK cell. From these lymphocytes, again, some activated effector cells are being produced. So what are these effector cells? So from B cell, you can see plasma cells are produced, and plasma cells are the cells which produce, anyone knows what is the role of plasma cell? Yeah, right, you are Shayan. Plasma cells produce antibody. Yes, Himapriya also, you are right. So plasma cells are the effector cells of the lymphocyte arm or the lymphoid lineage of the immune cell, which can produce antibodies. Now there are several types of activated T cells also. So as we learn, as we go through this course, we will learn in the later weeks that T cells can be cytotoxic T cells. So the effector T cells, the T cells which will produce some function in the immune system to eliminate the pathogen. So they will be either effect helper T cells or cytotoxic T cells. And also these NK cells or innate lymphoid cells, these cells also mature more and more in the lymph nodes so as to become effective, right? If this part is clear, we will now move on to what happens during an infection. So I guess you all are very curious to know that if the immune system is being able to protect our body from different kinds of infections, and as we know that there are many pathogens which are there in our environment, and if all these pathogens or many of the pathogens infect us or try to infect us, what is the role of immune system, or what is the first response of the immune system? Now you might have heard about the innate arm of the immune system and the adaptive arm of the immune system, right? So the innate arm of the immune system is the arm of the immune system which actually becomes active first when there is an infection. So there are some anatomical and chemical barriers on our body, like for example our skin and the mucosal epithelium and like that. So when the pathogen is being able to cross, for example our skin, so then what happens, the innate arm of the immune system gets activated. And what happens in this type of system, you can see that when suppose these red colored they are bacteria, these red colored cartoons are bacteria, and these bacteria are suppose they have gone into one of our tissues, and these cell, this is macrophage, right? So this macrophage, what it does is that it tries to engulf the bacteria, right? And when it engulfs the bacteria, so the first action is that it releases some chemicals called cytokines and chemokines. So in the later classes we will learn more about the cytokines and chemokines, what they are, what are their different classes. But just to introduce you to them, so they are different chemicals which help the immune cells either to activate some other cells, right? So suppose I am undergoing infection, please come and help me, that kind of response is provided by the cytokines. Or the chemokines are the type of molecules, the chemoattractants they are. So when suppose there is an infection, these chemokines are released by some of the cells, and then these chemokines attract more and more immune cells to the site of infection. All right. So here we can observe it that the macrophage is engulfing the bacteria and producing these molecules, chemokines and cytokines. And what is the effect of these molecules? You can see here vasodilation and increased vascular permeability. Hello? Are you asking anything? Hello? Okay, all right. So what is vasodilation? So this is a blood vessel, as we can see. This is a blood vessel, and there are many blood cells which are inside the blood vessel. And what happens is that due to the action of this particular type of cytokines, the endothelium cell the endothelial cell which line the blood vessels, they are dilated in the sense that the gap between these endothelial cells, as you can see here, from here there is almost no gap, but here the gaps between these endothelial cells increase, right? And because of which the vascular permeability, the permeability of the blood vessels increase, right? And because of which the blood cells, the immune cells which were circulating in blood so far, can come to the site of infection. So this macrophage is producing these chemokines and cytokines which are calling in more and more immune cells to the site of infection, right? And this neutrophil, as you can see, this is trying to cross the blood vessel and coming to the tissues. And because of this, what happens is that it causes inflammation. So inflammation is something which we very commonly see. Suppose there's a cut on your skin, and when there is a cut on your skin, you can see after some time it will become red, and it will swell, and it will become heated up, right? So that kind of these are actually the characteristics of inflammation. So you can see redness, heat, swelling, and also pain. So these are the few characteristics of inflammation by which we can understand that the immune cells are now coming to the site of infection. Suppose there is a cut and there has been a entry of some bacteria, right? Because I told you that the skin is the protective barrier against many pathogens, right? But if there's a cut, if there's a wound on the skin, so the bacteria will, or the other pathogens will try to invade your body, your system, through that wound. And in that case, the macrophages and other cells, I will discuss them later on, which are there on the tissue, will try to protect us in this way. So they will produce these chemokines and cytokines, and they will call in more and more immune cells, which are neutrophils and dendritic cells and these cells. And then when they will come, it will cause inflammation, and so it causes redness, heat, and swelling and pain. And then these inflammatory cells, when they come into this tissue, when they migrate into this tissue, it releases some mediators which will cause pain also. And this is actually one of the first responses when infection happens inside our body. So this is one of the innate immune responses which occur, right? And so here I will just introduce you to some of the cells which are crucial for this kind of innate immune response. So what are the innate cells of our body? First is the macrophage, which I was talking about so far. And macrophage causes phagocytosis and activation of bactericidal mechanisms, and it also presents antigen. So antigen presentation is one of the crucial properties of these immune cells, and most importantly that they can present these antigens to the cells, which will actually initiate the adaptive immune response of our body. So if our innate immune system cannot handle whatever is going on, when the pathogen is infecting our body, if the innate immunity cannot handle the particular pathogen, then of course it will allow the adaptive arm to become activated, and then finally eliminate the pathogen from our system. So these cells are macrophage, dendritic cell. Dendritic cell, as you will learn later on, that it is the linkage between innate and adaptive immune system. Then neutrophils. Neutrophils are the cells which come in more and more amount to the site of infection. Then eosinophil, basophil, mast cell, these are the granulocytes. You may be familiar with these type of cells, and they are important for providing antiparasitic immunity. both eosinophil and basophil, right? Okay, so now coming to the adaptive immunity. So so the adaptive immunity is basically provided by the lymphocytes, and as we know, there are two types of lymphocytes: B cells and T cells, right? And these types of cells are very important because they can recognize an antigen and produce a receptor, or they have specific receptors with which they can specifically recognize particular antigens, right? So they have specificity, which was lacking in case of the innate immune arm. I'm not telling that the innate immune system is non-specific. They also have certain level of specificity, but not as much as the adaptive immune arm, right? So the B cells are very crucial for adaptive immune system because they produce these antibody molecules, right? And antibody molecules have this kind of a structure. So here you can see that the antibody molecules have two chains. One is this green one, right? So this is a heavy chain, and there is another chain which is the short one, which is called the light chain of the antibody. And there are usually, you can see in this structure that there are two heavy chains in this antibody and two light chains which are joined together, joined to each other, and forms this Y kind of a structure. And it is also essential to remember that these heavy chains and light chains, they have a region which is constant. So it is constant in different types of antibodies, and a variable region. So this variable region is important for antigen binding. So this variable region forms this antigen binding site with which it can recognize any kind of chemicals. So antibodies can actually potentially like recognize any types of chemical structures, be it proteins or be it any other types of structures. Whereas so this is the antibody which are produced by B cells, or the activated B cells, which are plasma cells. Whereas the T cell receptor is quite different from these antibody molecules, because the T cell receptor has a so they are these receptors which are attached to the membrane of the T cells, whereas antibodies are not membrane bound, right? So T cell receptor, they also have two chains. Here you can see alpha and the beta chain, and they also have this variable region which functions as the antigen binding site. Yes, so Shayon, to answer your question, he's actually asking like what kind of bonds are there between the heavy and light chains. Yes, disulfide linkage is also there. There is hydrogen bonding also. So I'm just briefly telling you here because there will be classes where we will be discussing antibody structure in detail. So yes, disulfide linkages join these two chains. You're right. Right, and the antigen binding site of the T cell receptor is also the variable region, whereas the constant region is constant for all kinds of T cell receptors. Right, so these are the two weapons of the adaptive immunity. One is the antibody and another is the T cell receptor. Right, and this is a typical diagram of how an antibody can recognize an antigen. So this is one antigen molecule, as you can see, and here it is a protein antigen, and you can see there are particular regions in this protein antigen, here shown in blue, which acts as the epitope And what is this epitope? The epitope is the region of the antibody which the epitope is the region of the antigen which can be recognized by the antibody, right? And in this antigen antibody reaction, this antigen antibody reaction is very crucial, because antibodies, as I told you, that they can they are soluble, so they are not membrane bound, so they can practically recognize any and every antigen which is there in the extracellular space, right? And they do not also need any other molecule to recognize the antigen. So why am I saying any other molecule? Because we will see that in case of a T cell receptor. So a T cell receptor binds to antigen via MHC molecule. MHC is what? MHC is major histocompatibility complex, right? And we will see that a T cell receptor actually cannot bind to antigen directly, like we saw in case of an antigen antibody reaction, right? But the T cell receptor requires the mediation by this major histocompatibility complex proteins, or the glycoproteins. So these are glycoproteins. And remember one thing, these MHC molecules actually present a part of the antigen processed. So it presents a processed antigen to the T cell receptor. So what happens, you can see here in this diagram. So one thing just to mention, that these all diagrams are available in the Genway Immunobiology book. You can check also. It's in the first chapter of the book, right? So the epitopes which the T cell receptor can actually recognize, the epitopes on the antigen surfaces which the T cell receptor can recognize, are often buried in the protein. So this is a three-dimensional model of the protein, and this red portion is actually the epitope, right? And this can remain buried inside the protein, so it might not essentially be exposed to the surface of the protein. But what happens if the antigen has to be processed? This is called antigen processing. It occurs inside the cytoplasm, of a cell, and so the antigen is first broken down. So it is chopped up, right? And the peptide fragments are generated, and these peptide fragments are then presented on these MHC molecules. So you can see that on the MHC molecule there is this groove kind of a structure on the MHC molecule, right? And on this groove, this epitope peptide can sit, right, on the cell membrane. And so suppose this cell is what? This cell is actually a APC, This cell is actually a APC. APC is nothing but antigen presenting cell. Right? So this is an APC, and this is presenting the antigen, the epitope peptide. Yes, to answer your question, there is a difference between a TLR and TCR. This TLR is actually the pattern recognition receptor. Just a minute. TLR is a pattern recognition receptor, or often called PRR as in abbreviation. So TLR is nothing but no, they are not present on T cell. No, both of them are not present on T cell. TLR are present on the macrophages, right, and some of the antigen presenting cells. And remember, this is involved in the innate immune system. So these are pattern recognition receptors which can recognize PAMPs. What is PAMP? PAMP is pathogen associated molecular pattern. So suppose if I am being infected by a bacteria, there will be certain patterns on the bacterial surface which my body can recognize, right? And these are called the PAMPs. And the PAMPs are recognized by several receptors which are residing on our immune cells, right? For example, this macrophage have these TLRs which can recognize certain PAMPs or pathogen associated molecular patterns on the surface of the bacteria or any other kind of pathogens, and then they can actually engulf the pathogen and they can process it and they can kill the pathogen and they can eliminate the pathogen, right? So that is what happens in innate immune system. So this diagram that I had shown you, this engulfing of bacteria by the macrophage, this actually occurs through some receptors. So just like that, macrophage cannot go and engulf the bacteria. It has to recognize first that it is a bacteria and it is not some cells of the self, right? So how does this recognition take place? One of the receptors of this recognition is TLR, right? So that is not TCR. So TLR is involved in innate immune response and TCR is involved in adaptive immune response of our body and resides on the T cell, the latter. I hope I have cleared your doubt. Alright, so like I was telling you that this now when this epitope is presented on the MHC molecule, now these APCs are actually circulating in the blood, and then some T cell, so these T cell which recognizes this antigen, they are naive T cells. What do we mean by naive? Naive T cell is something which have not met the antigen yet, right? So naive T cell is something which is not which has not met antigen, and these naive T cells are circulating in the blood. And what happens when the MHC molecule presents some antigen, the T cell receptor comes and binds to the MHC antigen or MHC peptide complex, right? And then this kind of interaction between the MHC peptide complex and the TCR actually activates the T cell. So now the T cell is not naive anymore, so it becomes effector T cell, right? So I hope yes, Khushi, you're right. So antigen has to bind to the MHC first, or the antigen has to be processed inside the cell first and then bind to the MHC and then be presented on the surface of the APCs or antigen presenting cells, and then the TCR can actually recognize the antigen. The TCR will not be able to recognize antigen when it is like not bound to MHC, right? So MHCs are important in this case. Alright, so now you might have little bit understood the difference between an antigen antibody reaction and a TCR antigen reaction, which is mediated by these MHC molecules, right? Okay, so now I will just go through some questions. What is the difference between T cell, B cell and antibody? T cell and B cell, they are different types of lymphocytes, and the antibody is actually the molecule which is antibodies are actually the B cell receptors, the receptors on the B cells which are secreted by the B cells, and when they become soluble they are known as antibodies. And another question was there, is MHC and APC same? No, absolutely not. MHC is the major histocompatibility complex. it is actually a glycoprotein. So it is protein conjugated with carbohydrate, right? And it's there are 3 protein chains which make up this MHC complex. But APCs are antigen presenting cell, right? I've written it here. APC is an antigen presenting cell, so they will have MHC molecules which will help the antigen presenting cell to present its antigen. Alright? Okay, so I'll now move on to some of the questions. Okay, so please go through the question. It says that how many of the followings are correct with respect to antigen recognition by antibodies and or T cell receptor. So antibodies, T cell receptor, I was just discussing right now. So I hope you can take a guess. Number 1 is both can recognize any type of chemical structure. What do you think? Like, I told you already that antibodies can recognize any type of chemical structures. So that is true for antibodies, but what do you think for T cell receptors? Is it true or is it false? Okay, next coming to option number 2, which is antibodies may recognize any type of chemical structure, but T cell receptors usually recognize protein antigens only. Okay, so you can see here that option 1 and 2, they are mutually exclusive. Okay, somebody has answered. All right, we will check your answer. Okay, so yeah, so 1 and 2 are mutually exclusive, so only one of them will be correct. Right? And here we can see that in these options, so it's better to check the options always before solving the MCQs so that you get a clear understanding. And in the options you can see that option A and D, they have 1. Option D has both option 1, the statements 1 and 2, right? Yes, so if statement 1 and 2 together cannot be correct because they are just opposite statements. Okay, Sandeep is saying TCR can bind only specific chemical. Alright, we will see. Next, coming to the option 3, which is TCR interact with antigen presented by MHCs but not with free antigen. So this I was discussing so far. You might know that this is true, right? TCR can only interact with antigen when it is presented by MHC. And the last is antibodies can directly recognize the epitopes of native antigen in the serum or in the extracellular spaces. So option 4, you can see that the native antigen, the antigen which is, yes, some are answering. I'll request all of you to, if you have any guess at this answer, please write it in the chat box. Alright, so antibodies can recognize the native antigen. Here native means that the antigen is not processed. And this is something that we saw in the previous, this here, that this antigen, this protein antigen is not processed. It is a native, it is in its native form, that is it is in its 3D form. Whereas the TCR requires processed antigen, right? So here we can say here, therefore, option 3 and 4, they are correct, right? So whether will it be 1 and 2 are not correct, so option D is cancelled. Whether will it be only 3 and 4 or 2, 3 and 4 or 1, 3 and 4? And the correct answer is B. So all of you who were writing solution is B, you are correct. Yes, option A is like only the first statement is wrong. Only the first statement is wrong, that is both can recognize any type of chemical structure, that is wrong. So TCRs can only recognize protein antigens only, whereas for antibodies it is possible to recognize any type of chemical structure. So those who have answered B, you are correct. Okay, next we will move on to another question, question number 2. which is a very straightforward question. I hope you can all answer this. Please write your answers in the chat box. Yes, somebody is saying A, C, A, A, C, A, C, C, B, C, A. All right, all right. Okay, I can clearly see there is like the answers are quite divided between A and C. So 50% are saying A, 50% are saying C. Some of you are saying B, only thymus. Okay. All right. All right. So if we go back to the first slides, the question is where is the maturation of T cells take place? And here you can see this, that the T cells, so these are the T cells, right? And the T cells, first, all of these cells, just give me a minute. All right, so all of these cells, immune cells, first originate in the bone marrow, I told you, right? And the origin of the T cells is obviously in the bone marrow, right? And then the T cell reaches thymus. So many of you were answering thymus, right? So then when the T cell reaches the thymus, it actually differentiates and it matures. So the right answer will be C. All right. So the origin of the T lymphocytes take place in bone marrow, whereas the maturation of the T lymphocytes take place in thymus. All right. So these two options are incomplete, and the origin takes place. So therefore, goes to option C, which is the site of origin of T cells, and maturation of T cells is thymus, right? All right. So here comes the solution, which is bone marrow and thymus respectively. All right, moving on to the next question. Before moving on to the next question, I'll just describe some few more concepts with you, which is one of them is antibody effector functions. So so far I was telling you that the T cell receptor and the antibody, these are the arms of the adaptive immune system, right? And I also showed you how the antigen can recognize the antibody, right? Sorry, this is not a question. You don't need to type anything. This is not a question. This is something I'm just going to explain to you. That is the effector functions of the antibody. So this effector function of the antibody is usually mediated by the Fc region or the constant region of the antibody, right? Were you saying anything, Arun? Okay. So the effector functions of the antibody are mediated by the Fc region or the constant region. I told you there are two regions on the antibody. One is a variable region and this constant region. So constant region brings out the effector functions, that is, after the antigen recognizes the antibody, next what will be the outcome of this recognition, how the antibody will eliminate the antigen. So this process takes place in three ways: neutralization, opsonization and complement activation. So these are the three types of effector functions that antibody can do, right? And here I will just go through very briefly one component of this antibody effector function, which is opsonization. So suppose there are specific antibodies, right? These are the specific antibodies, and there is a bacteria which has invaded our body, right? So there is a bacteria in the extracellular space. Now what happens, these specific antibodies, suppose they can recognize this bacteria, right? So there are specific epitopes on the bacterial surface which can be recognized by these specific antibodies. And then these antibodies, what they do is that they can coat this bacteria. So this is the bacteria, and these are being coated by many antibody molecules which are specific to this bacteria, right? So to the epitope that is present on the bacterial surface. And now these antibody are coating this surface of the bacteria, and next what they will do is that this type of coating of the bacteria by the antibodies will allow the macrophages to engulf and ingest this complex, the antigen antibody complex. So how does that take place? So these Fc region of the antibody is recognized by specific receptors which are called Fc receptors, right? Fc receptors are the type of receptors which are present on the macrophages and other types of cells, and they can recognize or they can interact with the Fc region or the constant region of the antibody, right? And then what will happen is that when the macrophage can, through these Fc receptors, so these yellow, they are the Fc receptors, right? So when they recognize these antigen antibody complex through the help of these Fc receptors, they can engulf it. So it is phagocytosed inside the macrophage, and then this macrophage kills the antigen antibody complex, and in that way the pathogen is eliminated from our body. So one of the major and effective functions of antibody is opsonization. There are two others, but neutralization. In case of neutralization, the similar kind of things happen, that the antigen antibody complex forms, and then it is digested by the macrophages. And in case of complement activation, what happens? Complement are special type of proteins in the immune system. So when the antigen is suppose coated by antibodies, and this coating of the antigen by the antibody creates or actually activates several complement proteins. And what happens, like it forms as a cascade. So it's like one after another, many complement proteins are activated. Shayon, can you tell me, can you ask the question instead of writing? So I'm not being able to understand. Yeah, difference between neutralization and opsonization is that neutralization takes place for toxins, right? But in case of opsonization, the whole antigen is actually engulfed. And in this complement activation, the different complement proteins are activated, which on one hand it is phagocytosed by macrophages, right? The antigen antibody complex, it helps in phagocytosis of the antigen antibody complex. Or it can also what it can also create is that it can create pores on the bacterial membrane, right? The complement proteins, when they become activated, they can create pores on the bacterial membrane, and then they can kill the bacteria. So these are some of the effective functions. And this Fc region is present usually on the antibody, of course, always it is present, but the macrophages can only interact with these antibodies when they are bound to antigen. All right, so when they are bound to antigen, then only this opsonization can take place. All right. Yes, antibody effector means that so when the antigen antibody complex is formed, what happens after that? That is the effective function of the antibody. So what does antibody do? That is its effective function, right? Okay, so this is one question I've given for you, which is Antibody first binds to antigens on the bacterial cell through variable regions. Then the antibody's Fc region binds to Fc receptors expressed by the macrophages and other phagocytes, facilitating phagocytosis. This process is known as neutralization, opsonization, complement activation, or co-stimulation. So, yeah, you know the answer, right? It is opsonization, right? Very good. Yeah, all of you are giving correct answer. So this process is actually opsonization. So B is the solution, right? Okay. The penultimate question I have for you today is the examples of primary lymphoid organs. So this is one straightforward question. There is nothing too much to describe here. Can you please write the answer? Okay, many of you are saying A. So what are the primary lymphoid organs? Yes, bone marrow is of course the primary lymphoid organ. Thymus is also primary lymphoid organ. What about spleen and lymph node? Does anyone know what is spleen and like what type of lymphoid organs are spleen and lymph node? Okay, Sandeep is writing don't know about bursa fabriceus, right? I'll explain that to you. Yeah, right. So some of you are writing spleen and lymph node are secondary lymphoid organs, you're right, or peripheral lymphoid organs also, right. So spleen, lymph node, malt. Any idea what is malt? Yes, mucosa associated lymphoid tissue. You're very right. Yeah, good. This is this malt or the mucosa associated lymphoid tissue. This is actually some patches on the mucosal surface which are rich in immune cells, right? So that also comes under peripheral lymphoid organ or secondary lymphoid organ. Whereas bone marrow, thymus and bursa fabriceus, they are the primary lymphoid organs. So your answer is correct. This is solution A, and the bursa fabriceus is actually present in birds. Alright, so our solution is A, bone marrow, thymus and bursa fabriceus. This was one quite straightforward question. Alright, and the last concept for today's class is MHC class 1 and class 2. So MHC molecules I was discussing previously, that these are the molecules which are the mediators which are helpful, which help the antigen to be presented to the T cell receptors, right? So there are two classes of MHC molecules, class 1 and class 2. Now the MHC class 1 molecules are the type of molecules which help in presenting certain types of antigen, and class 2 is different in that case. So that is only the difference between the MHC class 1 and class 2. They have slightly different structure as well, but I will not go into that detail now. But MHC class 1 and class 2, these are important because they process different types of antigens. So what are these different types of antigens? So suppose one is intracellular, another is extracellular. These are antigens, intracellular antigen and extracellular antigen, right? So what do I mean by intracellular and extracellular antigen? So suppose there is a virus, right? Virus, as you all know, currently also we are going through this pandemic of COVID virus, right? Coronavirus. So virus is one crucial pathogen, like a very deadly pathogen also. And what happens, virus, as we know that they are um, non-functional, or they're non-living outside the host, right? But once they invade the host system, and once they are into the cell, so once they invade inside the cell, they start producing their proteins, right? They start replication and they start producing their proteins. And so that is why viral antigens are always intracellular antigens, because the virus has invaded into the cell and now it is producing proteins. And MHC class 1 molecules actually present this type of intracellular antigens, right? So they process this kind of, so what happens is that the virus, when it goes into the virus, when it starts producing its proteins, these viral proteins are synthesized inside the cytoplasm of the host, right? And when these viral proteins are synthesized, they are also chopped off and they are processed and they are presented with the help of these MHC class 1 molecules. And then what does it do? This MHC class 1 molecule now presents its antigen to cytotoxic T cells. So I told you previously also that T cells, the effector T cells can be cytotoxic T cells or helper T cells. So they are different. We will in later classes actually discuss about how they are different, how their functions are different. But just to brief you here, cytotoxic T cells are the cells which can kill the pathogen infected cells, right? So that is why they are called cytotoxic, because they are killer T cells, right? And these MHC class 1 molecule presents its antigen to the cytotoxic T cells, and these cytotoxic T cells help to kill this virus infected cell. Okay, so that is about the MHC class 1. And what about the MHC class 2? The MHC class 2 actually presents its extracellular antigens to the T cells. And here you can see that what is extracellular antigen. Suppose there's a bacteria which is there in the extracellular space, right, which is outside the cells. And when this bacteria infects, there are macrophages residing in our tissues, right? These macrophages go and engulf this bacteria, right, like you have seen, you can see here, and this causes phagocytosis. The macrophages, they cause phagocytosis of the antigen, right, and it forms phagolysosome. Phagolysosome is what? It is the fusion of the phagosome with the lysosome, and it so these are these vesicles of phagolysosome, right, and which actually process these types of antigens, and then these antigens are presented by MHC class 2, right? So MHC class 2 can present extracellular antigen to the T cells. So here you can see T helper cells, Th1. So T helper cells are, again, there are different types of T helper cells. Here it is shown in this diagram, it is Th1. There are other types of T helper cells. We will discuss later on about them. But here, just to give you this introduction, this MHC class 2 is presenting this antigen to the Th1 cell, right? So that is how MHC class 1 and class 2 interact with the TCRs, respective TCRs. And just, this is the last question of today's session. Can you please, yes, CD4 and CD8. So these are cluster of differentiations. So CD4 is expressed by MHC class 2, like CD4 actually interacts with MHC class 2, and so CD4 and CD8, they are actually present on the T cell, right, and they help in this TCR MHC interaction. So they are kind of, we can say, helper receptors, right? So which help in this kind of TCR MHC interaction. And CD4 is present on the CD4 is present on the T helper cells, whereas CD8 is present on the cytotoxic T cells. And CD4 helps to interact with the MHC class 2 molecules, and CD8 helps to interact with the MHC class 1 molecules. I hope that answers your questions. Yes, many of you are answering. Right. Many of you have answered D, A, C, D. Clearly, there is no consciences in this question. Any other answers? Okay, so if there is no other answer, I'll just yes, A. Okay, okay, I'll tell you the correct answer. The correct answer is A, cytosol. And why is it so? Because the MHC class 1 molecules, I told you, they always process intracellular antigens, right? So these are the antigens which are, you can see, these are the particular type of virus, suppose, which has infected the cell, and these viral proteins are produced in the cytosol, right? And when these viral proteins are produced, they also are chopped off, and they are accompanied with MHC class 1 molecules, and they are presented on the surface. So these MHC class 1 molecules, they present the antigen which are derived from the proteins in the cytosol, right? Because the viral proteins are being produced. So like, suppose there are these ribosomes, and this is the viral mRNA. Right? Which are synthesizing the proteins. These are the viral proteins. So they come in the cytosol of the host cell, right? This is all happening in the cytosol of host. And these antigens are then processed and presented by MHC class 1. So that is why this is the answer. Why not Golgi bodies? Why not ER? Because these proteins are present in the cytosol only, and not in any other cell organelle. So anybody having any doubt? Any other doubt?
Yeah, actually, I want to know about the program specific. Like, do we need to attend both Friday and this Tuesday class, or only one is sufficient?
So, what do you think? Like, you have attended the Friday session as well, right? So, do you think my session and her session?
I did not.
Okay.
I did not attend it. So that's why I got actually confused, because I got 2 mails. So I thought Friday one is cancelled, and then Tuesday
Yes,
one.
ma'am, it's my question too. Like, I was also confused because I got 2 mails. Then I didn't attend the Friday's class session, and
Right.
it's my first class.
Yeah, so actually, you see, these live sessions are designed so that we can help you guys to understand this course in more detail. Like, obviously, professors are teaching you in the video lectures, but there is like little bit of interactive session is not there between you and professors, right? So to fill in that gap, actually, we are coming in to help you understand these courses more and more. And here we will be actually, what we are actually doing, so since there are 2 TAs, so we will be every day solving some questions with you, and actually going through some concepts which you have learned in this particular week and so on. So I would actually recommend that if you join both the live sessions, it will be good for you only, because you will be solving some more questions. So these are the questions, these types of questions also you will get in the assignment, right? So the assignment and the exam also, like final exam when you will face, so these kind of questions will come, and if you can solve with me, it will be a good thing for you. So I hope that answers your question, right?
Yeah, okay. So now two topic based question actually. So one is the guy, somebody asked, I think, CD4 and CD8.
Yes.
So is this a T cell or it's a different cell which is helping T cell?
No, actually, CD is called cluster of differentiation. So these are like some cell surface molecules. Don't worry, we will go in the detail of it, and in the lectures also, in the next coming weeks NPTEL lectures also, you will go through this CD. That will obviously answer all of your questions, but just to introduce you, so these are some cell surface molecules which are present on the T cell, right? And what do? They help the TCR to interact with the MHC. So in this TCR MHC interaction, so there are several components which play important roles in this TCR MHC interaction, and so one of them is these CD molecules, right? So there are CD molecules on the T cell, as well as there are CD molecules on the APCs which interact with each other, and they strengthen this interaction between TCR and MHC, right? And CD4 and CD8, right? CD4, as I already mentioned, I'm telling you just once more, that is CD4 is CD4 is present on the T helper cells, and they help the TCR to interact with the MHC class 2. Alright, and whereas CD8 is unique to cytotoxic T cells, right? And they help, since they are present on the cytotoxic T cells, and cytotoxic T cells interact with via the MHC class 1 molecules, right? So they the antigen presentation occurs via the class 1 MHC molecules. So CD8 actually helps in interaction with class 1 MHC molecule. So
Okay, thank you.
Yeah.
So another question is in a conceptual basis, like let's say if some other virus or bacteria comes which our innate system cannot able to fight,
Yeah.
as well as we first time we are recognizing that, so
Right.
there is no memory for it.
Yeah.
So is it T or B cell is able to handle that situation, or we really have to suffer for it to get over on this?
Actually, see, if any infection takes place in our body, so first the innate arm of the immune system comes and gets into the action, right? It tries to protect the body in whichever way is possible, and it fights with the pathogen. But however, it can be less powerful than the pathogen which has infected us, right? So pathogen might sometimes overcome the innate immune system, and in that case, what happens, the adaptive arm of the immune system gets functional, and you will be surprised to know that the adaptive immune system, the adaptive immune response can actually continue for days to weeks to months, right? So there are many responses which are categorized by this adaptive immune system, and they take place for days and months of time, days and weeks of time, and in that time the body tries to clear off the pathogen. However, like if you are trying to know that like if we take medicines, what happens, right? So in that case, the medicines also help us to clear off the antigen. If the body alone is not being able to clear off it in a, like what to say, like soon, then we take medicines also, right? So that also helps us, but our body will be able to fight it as long as it can, right? But because obviously you know that all diseases cannot be um, cleared off or eliminated by our bodies, right?
Yeah, so if there is no memory, still our body will try to fight it some or other way because it's so specific. So I'm just thinking, if it is so specific and first time we are experiencing, then how like we react about it.
Yeah, so actually, when the first time the infection is taking place, the first the innate immune system is being involved. If it is not being able to clear off, it is involving the adaptive immune system, and then the after quite some time, like after days or after weeks, what happens that the adaptive immune memory, the immune memory is being formed, right? And then this happens for the first time, when the first time the infection takes place, and suppose it is continuing for say 15 days or 20 days, the infection may continue, right? But when this pathogen will infect us in the next time, or suppose in the next year, or in after some like 5 years, suppose, if this memory, adaptive immune memory, still persists, then the memories will be able to clear off the infection in suppose 5 days, right? So you may understand what is the difference between the first time infection and when the memory is being um, helpful to clear off the pathogen in very very short time.
Okay, thank you, Surasree.
Okay, thank you. Any other doubts anyone has? Yeah, Shivam, please. Okay, so you are asking like MHC class 2 molecules. Yes, so MHC class 2 molecules, they present antigen which are derived from the chopped off proteins from the vesicles, the lysosomes and all. Like I was mentioning here, the phagolysosome that forms. Is that all? Okay, if there are no other questions, then I'll conclude today's session, and thank you all for joining today. We will be next meeting again the same time, same day, Tuesday, 7 to 8 p.m. Thank you all for joining. Bye bye.